Alright children, now we begin with the second phase of meiotic cell division, which is also known as meiosis 2, right? So we will talk about meiosis 2, which is a homotypic division. This is also known as equational division, and it is similar to mitosis, right? Again, meiosis 2 is divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. So that's in chronological order: prophase, metaphase, anaphase, telophase. And since this is the second phase of meiosis, so it is prophase 2, metaphase 2, anaphase 2 and telophase 2. Now if we go back to meiosis 1, we saw that the last phase of meiosis 1 was telophase 1, where regrouping of chromosomes had taken place, right, at the poles, right? Each pole received 1/2 of the chromosome. So now if we look at this figure over here, we see that this is the stage where telophase 1 has taken place, and the chromosomes have moved, 1/2 of the chromosome has moved towards the either pole, right? And from the original number of chromosomes, the chromosomes have become to half, right? So here the cleavage furrow, you can see, is formed, right? And now from this stage, what happens is interkinesis is a very short-lived phase. From here the cell undergoes meiosis 2, right? So meiosis 2 starts with prophase 2. You can see how the cell has divided here and gone into the next stage, that is prophase 2. Now in this stage, if we look at the individual figures, we see that chromosomes become visible along with their chromatids, right? So this stage immediately takes place after cytokinesis, right, and usually before the chromosomes are fully elongated. But in contrast to meiosis 1, meiosis 2 resembles a normal mitosis, as I told you, right? So in prophase 1, what happens? The nuclear membrane disappears, spindle fibers are formed. So if you look at the figure here, you can see how these spindle fibers are being formed, right? So nuclear membrane disappears, spindle fibers are formed, and the chromatids of a chromosome look X-shaped, attached only by their centromeres to the spindle fiber. Now if we look at the individual figure here, you can see the disappearance of the nuclear membrane, right? The nuclear membrane disappears. You can see spindle fibers will be formed over here in prophase 2, and the chromosomes, if you look at them, they look X-shaped and they remain attached with the help of their centromeres. So here are the two cells haploid having half the number of chromosomes, right? So that enter the next stage, that is prophase, right? So after prophase, the prophase is followed by another stage which is known as metaphase, right? So this is meiosis 2, so metaphase 2. Now if we look at the figure here of metaphase 2, we see the chromosomes, they are attached to the spindle fibers. Now here they are very clearly visible. You can see these spindle fibers here, these are the spindle fibers, right? And you can see how the chromosome is attached to the spindle fiber along the equator. This is the equator. So always in metaphase, on the equatorial plate, you see the chromosomes are attached, right? So they attach along with their centromere on the spindle fiber, right? So here you can see both the cells, right, undergoing metaphase, and in both you can see how the chromosomes, they attach on the equatorial line, right? And if you look at the chromosomes, you'll find that each chromosome consists of two chromatids joined by a common centromere and two chromatids, right? You can see crossing over has also taken place already in the previous stages. So you can see those exchanged pieces. The progression of meiosis in vertebrate oocyte stops at metaphase 2 stage, right? But what you need to know is that the chromosomes, they arrange or they align at the equator, and the microtubules from opposite poles of the spindle fiber, they get attached to the kinetochores, that is these structures over here, right? So that is metaphase 2, and you can see here very nicely they've mentioned chromosomes line up separately on the equator of the spindle and kinetochores pointing towards the opposite poles, right? Next stage now you will see is the anaphase stage, and at this stage here, this is the anaphase stage. It begins with the simultaneous splitting of the centromeres. Simultaneous, you can see both here also in this cell and in this also, splitting of the chromosome is taking place with the help of the centromere, right? So centromeres now in anaphase 2, right? It was different in meiosis 1, right? So in meiosis 2, anaphase 2, centromeres divide and the chromatids are separated. See, if you compare it, here they are joined together and here you can see they are separated like this. They are separated, right? The chromatids, they begin to move towards the opposite poles, right? You can see they are moving apart and they are coming towards the opposite poles, right? Similarly here also they are moving apart and they are coming to the opposite poles, right? They are being pulled. How are they moving? Is because the spindle fibers, if you look at the spindle fibers, the central region, if you see of the spindle fibers, you will see that the spindle fibers are breaking, right? Or they are contracting, right? And they are moving towards the pole. So this, since in the center only on the equatorial plate, the chromosomes in the metaphase stage were arranged. So since it starts breaking from this region only, so the centromere also splits apart from that region, and along with the spindle fibers they move towards the opposite poles, right? The last stage is the telophase stage, right? So the telophase stage is here, the telophase 2 stage. Following the arrival of the haploid number of daughter cells, chromosomes at the poles, they become thin, they become long, and the nuclear membrane reappears, right? If you look at it here, the nuclear membrane reappears, right? And the nucleoli will be reconstituted, means it will be made again. So at the end of meiosis, from each parent cell 4 daughter cells are formed, right? So there are 4 daughter cells that are formed, and if we look at the compiled diagram here, you can see these are haploid cells that will lead to the formation of 4 daughter cells, right? So this leads to the formation of 4 daughter cells in meiosis 2, right? So now if we talk about the significance or the importance of meiosis, meiosis is a mechanism by which conservation of specific chromosome number, we can retain the specific chromosome number of each species. For example, in every generation all the human beings have 46 chromosomes, right? So how can this be retained? Only when this is turned into haploid, and when these gametes, haploid gametes, when they fuse again, 46 chromosomes are formed. That is what we've learnt, right? So it is this cell division, meiosis, that helps to retain the number of chromosomes in each species, right? This is achieved across generations in sexually reproducing organisms. Even though the process results in reduction of chromosome number by half, it also increases the genetic variability. Why? Because you can see parts or segments of the chromosomes, paternal and maternal, they're exchanging from each other. That is how we are different. That leads to variation in the species. That is how the next generation is different from the previous generation. And variations are very important for the process of evolution, right? So this variation is important, as I told you, because of crossing over, the interchange of segments. They enable exchange and recombination of parental characteristics. That is why offsprings, you see, the offsprings, all of us, we resemble both our parents in some or the other way. So that is because of crossing over that has taken place at the time of meiosis. Now, at the same time, if we talk about the significance of mitosis, right, we studied mitosis also. If we talk about the significance of mitosis, what is the importance of mitosis? It ensures that both daughter cells, they receive identical and complete set of chromosomes. Each set of chromosomes is uniquely characteristic of a particular species. The cells that make up the body but not involved in sexual reproduction all undergo mitosis, right? This aids simpler organisms like bacteria to reproduce asexually. It allows regeneration of damaged or worn out body tissues. Remember, when we get hurt or there's a cut somewhere, that gets repaired, right? So cell repair also is one of the significance of mitosis. Growth that takes place, cell growth, we grow in size, that is also the result of mitosis. So these are the basic significances of mitosis, right? So significance of mitosis and meiosis is asked, differences are asked, right? Then the various stages you can be asked to explain. So that's about it for today's class.